Hello everyone. I will repeat the Hello everyone. I will repeat this question number 27. In a zygote, there are 24 pairs of chromosomes. Then the number of chromosomes present in gametes are option A 24 pairs, option B 48, option C 6 pairs, option D 24. Here the question is, in a zygote, there are 24 pairs of chromosomes. 24 pairs is nothing but 48 chromosomes are totally present in zygote. This zygote is formed by uh, combining both chromosomes from male and female, right? Male and female chromosomes to form the zygote. So totally 48 chromosomes are present in zygote. But the question is, the number of chromosomes present in gametes. So each gamete contains 24 chromosomes. Hence the option D is the correct answer. So the number of chromosomes present in gametes are 24. Thank you. Hello everyone. Let us see 13th main. Answer the following questions. Question number 32. Name the following. In that first one, structures that prevent the collapsing of air passage part of the trachea in humans is called what? Okay. The question is, structure that prevent the collapsing of air passage part in trachea. Air passage part in trachea dalli collapse ಆಗದೆ ಇರೋ ಹಾಗೆ ತಡೆಯೋ ಸ್ಟ್ರಕ್ಚರ್ ಯಾವುದು ಅಂತ ಕೇಳಿದ್ದಾರೆ. So the answer is rings of cartilage. See, this is the cartilage rings. This cartilage rings always helps to keep this air passage open. Hence, due to this rings of cartilage prevent the collapsing of air passage part of trachea in humans. So, 32 in that second, nitrogenous chemicals that are separated from the blood by the kidney, that is called. Okay. So, nitrogenous chemicals that are separated from the blood by the kidney. See, this is the picture of kidney. We know that what is the function of kidney and all, right? Kidney is in bean shaped organ. It is very important part in our body. The main function of kidney is it filters the blood, right? And again helps to excrete the waste things which is produced in our body. So here the question is, nitrogenous chemicals that are separated from the blood by the kidney. The answer is urea and uric acid. These are the nitrogenous chemicals that are separated from the blood by the kidney. Thank you. Hello everyone. Let us see question number 32. In that A, in which form the excess glucose is stored in plants and animals? This is the question. This is our question in question number 32. In that A, which form excess glucose is stored in plants and animals? Plants and animals alli excess glucose ಯಾವ ಫಾರ್ಮ್ ಅಲ್ಲಿ ಸ್ಟೋರ್ ಆಗುತ್ತೆ? That is the question. So, see, in plants, this excess glucose is stored in the form of starch. In plants, this excess glucose is stored in the form of starch, and in animals, this excess glucose is stored in the form of glycogen. See, in plants, this plants store excess glucose as complex carbohydrates like starch in various parts such as leaves, roots and seeds and all. Okay. So in animals, this excess glucose is stored in the form of glycogen. This glycogen primarily in liver and muscles in animals. So here the answer is the excess glucose is stored in plants is starch, and this excess glucose stored in animals is in the form of glycogen. Then question number 32 in that B question, name the structure of humans small intestine that absorb digested food. So here the question is which structure of the human's small intestine that helps to absorb the digested food. Digested food ಅನ್ನ ಅಬ್ಸಾರ್ಬ್ ಮಾಡೋದಿಕ್ಕೆ ಯಾವ ಸ್ಟ್ರಕ್ಚರ್ ಹೆಲ್ಪ್ ಮಾಡತ್ತೆ ನಮ್ಮ ಸ್ಮಾಲ್ ಇಂಟೆಸ್ಟೈನ್ ಅಲ್ಲಿ ಇರೋದು ಅನ್ನೋದು ಕ್ವಶನ್. ದ ಆನ್ಸರ್ ಈಸ್ ವಿಲ್ಲೈ. ಓಕೆ, ಸಿ ವಿಲ್ಲೈ ಈಸ್ ದಿ in our humans small intestine, it contains number of or thousands of small finger-like projections that is called villi. These villi help to increase the surface area to absorb the digested food. So, the structure in the human body of small intestine that absorb digested food is called villi. So, villi is the answer. Thank you. Hello everyone. Let us see question number 33. Why does the stem of a growing plant bend towards the light? Explain. Hello everyone. I will repeat this question. Question number 33. Why does the stem of a growing plant bend towards the light? Explain. See, one type of hormone is generated in plants that is called auxin. This is the main reason for the stem of a growing plant which bend towards the light. So you have to write the explanation here. The explanation is like this. When growing plants detect light, a hormone called auxin, it's synthesized at the cells of a shoot top. Okay, see here, when the sunlight is detected from this plant, the hormone that is called auxin is generated in the plants. This is synthesized at the cells of the shoot top. When light is coming from one side of the plant, auxin diffuses towards the shady side of the shoot. This makes the stem of a growing plant bend towards the light. So when the sunlight is fall on this plant, the auxin hormone is generated. This light is coming from one side of the plant. This generated auxin hormone start to diffuse towards the shady side of the shoot. See, like this, auxin is generated and it starts diffusing towards here, shady part of the shoot. This makes the stem of a growing plant bend towards the light. This is the explanation. Thank you. Hello everyone. Let us see question number 33. Write any two differences between central nervous system and peripheral nervous system. Here you have to write the any two differences between the central nerve system and peripheral nerve system. Let us see what is the difference between this central nerve system and peripheral nerve system. See, first of all, the first difference is in central nervous system, it includes brain and spinal cord. So the central nervous system part are brain and spinal cord. These two parts comes under the central nervous system. In peripheral nervous system, it contains cranial nerves and spinal nerves. This peripheral nerve system contains the part cranial nerves and spinal nerve system. This is the first difference. And second difference, this central nerve system receive information from all parts of the body and integrate it. This is the function. This central nerve system receive information from all the parts of our body and then it get integrated. But in peripheral nerve system, this facilitates communication between the central nerve system and the rest of the body. This peripheral nerve system facilitates the communication between the central nerve system and the remaining part of our body. These two are the differences we can write about the central nerve system and peripheral nerve system. Any two differences. Okay. Thank you. Hello everyone. Let us see question number 34. In that A question, ozone layer is a boon to the life of the earth. Justify. So you have to justify your answer why the ozone layer is a boon to the life on the earth. Okay. See, we know that what is ozone layer, what is the function of ozone layer and all. Our sun, when the sunlight is coming from sun to earth, this sunlight contains so many harmful radiations like ultraviolet, visible and all. Okay, see, these rays are very dangerous. Once it directly reaches our earth, it causes some diseases like skin cancer, or it damages our eye, or it also harm to the crops also. So to prevent this direct uh, direct falling of this ultraviolet radiations, this ozone layer helps to prevent this radiation. ಸೂರ್ಯನಿಂದ ಬರ್ತಾ ಇರುವಂತಹ ಡೇಂಜರಸ್ ಕಿರಣಗಳಲ್ಲಿ ಒಂದಾದ ಯು ವಿ ರೇಡಿಯೇಷನ್ ಡೈರೆಕ್ಟ್ ಭೂಮಿಗೆ ತಲುಪಿದ್ರೆ ನಮ್ಗೆ ಹಲವಾರು ಡಿಸೀಸ್ ಅನ್ನ ಕೊಡತ್ತೆ. ಸೊ ಅದಕ್ಕಾಗಿ ಅದನ್ನ ಪ್ರಿವೆಂಟ್ ಮಾಡೋದು ಓಝೋನ್ ಲೇಯರ್. ಇದೇನ್ ಮಾಡತ್ತೆ, ಇಟ್ ಪ್ರಿವೆಂಟ್ಸ್ ಹಾರ್ಮ್ಫುಲ್ ಡಿಸೀಸಸ್ ಡೈರೆಕ್ಟ್ ಅರ್ತ್ಗೆ ರೀಚ್ ಆಗದೆ ಇರೋ ತರ ಓಝೋನ್ ಲೇಯರು ಅದನ್ನ ಪ್ರಿವೆಂಟ್ ಮಾಡುತ್ತೆ. ದಟ್ಸ್ ವೈ ವಿ ಕಾಲ್ಡ್ ಇಟ್ ಆಸ್ ಬೂನ್ ಟು ದಿ ಲೈಫ್ ಆನ್ ದ ಅರ್ತ್. ಸೊ the ozone layer is a boon to the life on earth by blocking harmful ultraviolet radiations or rays from the sun. The main function of this ozone layer is it prevent for this harmful ultraviolet radiation which comes to the earth. Okay, so that's why we consider ozone layer is a boon to the life on the earth. Then question number 34, in that B question, the organisms of which trophic level in a food chain are the most affected by biological magnification and why? The organisms of which trophic level in a food chain are the most affected? So this is the different trophic levels in food chain, right? See, it contains so many steps. In basic or first step, the producer ಅನ್ನೋದು ಮೊದಲ ಹಂತ. Here the food is generated, right? Producer ಅಂದ್ರೆ ಆಹಾರವನ್ನ ಪ್ರಿಪೇರ್ ಮಾಡೋರು. ಸೊ ಈ ಪ್ರೊಡ್ಯೂಸರ್ ಅನ್ನ ಅವಲಂಬಿಸಿಕೊಂಡು ಪ್ರೈಮರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ಆರ್ ದೇರ್. These primary consumers consume their foods in from the producers. So primary consumers ಏನ್ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ ಪ್ರೊಡ್ಯೂಸರ್ಸ್ ಅನ್ನ ತಿನ್ನೋದ್ರ ಮೂಲಕ ಆಹಾರವನ್ನ ಪಡ್ಕೊಳ್ತಾರೆ. Again the secondary consumers get their food from primary consumers. So this process continues. ಈ process ಏನಾಗ್ತಾ ಹೋಗುತ್ತೆ, ಕಂಟಿನ್ಯೂ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. In trophic level, the first level is producers and second one is primary consumers and third one is secondary consumers and fourth comes in tertiary consumers. These all are the different trophic levels in a food chain. The question is, which trophic level in this food chain are most affected by biological magnification? So the trophic level, organisms at higher trophic levels that consumes most affected biological magnifications. Because so ಇಲ್ಲಿ ಏನಾಗತ್ತೆ ಅಂದ್ರೆ ಪ್ರೊಡ್ಯೂಸರ್ಸ್ ಅನ್ನ ತಿಂದು ಪ್ರೈಮರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ಇದ್ರೆ ಪ್ರೈಮರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ಅನ್ನ ಸೆಕೆಂಡರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ತಿನ್ನತ್ತೆ, ಸೆಕೆಂಡರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ಅನ್ನ ಟರ್ಷರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ತಿನ್ನತ್ತೆ. ಸೊ ಈ ತರ ಮಾಡೋದ್ರಿಂದ ಪ್ರೊಡ್ಯೂಸರ್ಸ್ ಅಲ್ಲಿ ಇರುವ ಹಾರ್ಮ್ಫುಲ್ ಕಂಟೆಂಟ್ ಏನಿದೆಯೋ ಅದು ಪ್ರೈಮರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗತ್ತೆ. ಪ್ರೈಮರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ಅಲ್ಲಿ ಅವೆಲ್ಲ ಏನಾಯ್ತು, ಗ್ಯಾದರ್ ಆಗಿ ಪ್ರೈಮರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ಅನ್ನ ತಿನ್ನೋ ಸೆಕೆಂಡರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ಗೆ ಇದು ಟ್ರಾನ್ಸ್ಫರ್ ಆಯ್ತು. This process continuously takes place. Finally, the final level is tertiary. So organisms at higher trophic level, that is tertiary consumers, it consumes more affected by the biological magnification. So why? You have to write the reason for this. Why? Because okay, the accumulation of harmful chemicals increases as move to the next trophic level in a food chain. See, this producers ಅಲ್ಲಿ ಸ್ವಲ್ಪ ಅಮೌಂಟ್ ಅಲ್ಲಿ ಡೇಂಜರಸ್ ಥಿಂಗ್ಸ್ ಇದ್ರೆ ಅದನ್ನ ಯಾರು ತಿಂದು ಬೆಳಿತಾರೋ ಅವರಿಗೆ ಅದು ಟ್ರಾನ್ಸ್ಫರ್ ಆಗತ್ತೆ. ದಿಸ್ ಪ್ರೊಡ್ಯೂಸರ್ಸ್ ಇವರನ್ನ ಪ್ರೈಮರಿ ಕನ್ಸ್ಯೂಮರ್ಸ್ ಕನ್ಸ್ಯೂಮ್ ಮಾಡತ್ತೆ. ಸೊ ಹಿಯರ್ ಇಟ್ ಟ್ರಾನ್ಸ್ಫರ್ ಫ್ರಮ್ ಪ್ರೊಡ್ಯೂಸರ್ಸ್ ಟು ಪ್ರೈಮರಿ ಕನ್ಸ್ಯೂಮರ್ ಲೈಕ್ ದಿಸ್. ಇದೇ ತರ ಎಲ್ಲ ಲೆವೆಲ್ಸ್ ಅಲ್ಲಿ ಏನಾಗ್ತಾ ಹೋಗತ್ತೆ, ದಿಸ್ ಹಾರ್ಮ್ಫುಲ್ ಕೆಮಿಕಲ್ಸ್ ಕಂಟಿನ್ಯೂಸ್ಲಿ ಟ್ರಾನ್ಸ್ಫರ್ ಫ್ರಮ್ ಸ್ಟೆಪ್ ಬೈ ಸ್ಟೆಪ್. So that is the reason for the accumulation of harmful chemicals increases as move to the next trophic level in a food chain. This is the reason. Thank you. Hello everyone. Let us see question number 35. In that A, explain the determination of height of a plant by genes. How the height of a plant by genes is determined. That is the question. We know that plants ಅಲ್ಲಿ ಕೆಲವೊಂದಿಷ್ಟು ಜೀನ್ಸ್ ಇರುತ್ತೆ, ಅದು ಹೈಟ್ ಆಗಿ ಬೆಳೆಯೋದಕ್ಕೆ ಕಾರಣ. And again this plants contain some or it produces some hormones like gibberellins, right? Gibberellin is one type of hormone which is produced in plants. This is the main cause for the growth of a plant. So genes secretes a specific enzyme. If this enzyme works efficiently, the plant grows taller. That is the reason for the height of a plant by genes. So determination of height of a plant by genes. You have to explain this. First of all, genes secrete a specific enzyme. If suppose that enzyme works properly or efficiently, the plant grows taller. Then 35th question in that B, explain the determination of sex of the male child in humans. The determination of the sex of the male child in humans. You have to explain this. Okay. First of all, here we can see this male and female chromosomes. Okay. First thing is one odd pair of sex chromosomes found in father. They are X and Y chromosomes. See, this father contains odd pair of sex chromosomes, that is X and Y. X and Y are sex chromosomes in male. That is, so, but the mother has only X and X chromosomes. So female, it contain only X and X sex chromosomes. But in male, it contain X and Y odd pair of chromosomes. The child received X haploid chromosomes from father and also receives X chromosome from mother. Therefore, XX pair of chromosomes represents girl child. See, if suppose this child receives X from mother and again X from father. So X and X pair of chromosomes are form the zygote, right? This represents it is girl child. When X is coming from mother and X is coming from father. So X and X pair of chromosome represents girl child. If suppose the child received Y haploid chromosome from father and also receives X chromosome from mother, therefore X and Y pair of chromosome represents boy child. See, when the time of zygote formation, here the child receives Y haploid chromosome from father and X from mother. So finally X and Y pair of chromosome, it represents the boy child. So this is the way of determination of child. Okay. Thank you. Hello everyone. Let us see question number 35. A red flowering pea plant, capital RR, is crossed with a white flowering pea plant, that is small rr. Show the result of F2 generation with the help of a checkerboard. Write the phenotypic ratio and genotypic ratio of the plants obtained in F2 generation. So here the question is a red flowering pea plant, that is RR, is crossed with a white flowering pea plant, that is small r and small r. Okay. Then you have to calculate F2 generation with the help of a checkerboard. This and write the phenotypic and genotypic ratio of the plants obtained in F2 generation. See here, first you have to calculate F1 generation. Here, red flowering pea plant, capital R and capital R, crossed with white flowering pea plant, that is small r and small r. So it gives capital R and small r, right? It gives capital R and small r gametes. This is about the F1 generation. F1 generation. So by using this gametes, you have to find out the F2 generation. The question is find out the F2 generation, right? In first of all, you have to calculate F1 generation. First F1 generation gametes ಅನ್ನ ಕಂಡಿಡ್ಕೊಂಡು ನಂತರ F2 generation ಗೆ ಚೆಕ್ಕರ್ ಬೋರ್ಡ್ ಅನ್ನ ಹಾಕಬೇಕು. Here first we have to cross RR with small rr. Okay, ಇದನ್ನ ಕ್ರಾಸ್ ಮಾಡಿದ್ರೆ ಸಿಗೋದು R and R. Let us see how to form the checkerboard for F2 generation. So now you get the gametes, that is R and R, right? So after this F1 generation ಅಲ್ಲಿ R and R ಸಿಕ್ಕಿದೆ. ಈಗ ನೀವು ಇದನ್ನ ಇಟ್ಕೊಂಡು F2 generation ಅನ್ನ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡಬೇಕು. Here again cross this RR with RR. So RR ಅನ್ನ RR ಜೊತೆ ಕ್ರಾಸಿಂಗ್ ಮಾಡಬೇಕು. ಅದನ್ನೇ ನಾವಿಲ್ಲಿ ಚೆಕ್ಕರ್ ಬೋರ್ಡ್ ಅಲ್ಲಿ ತೋರಿಸಿದ್ದೇವೆ. ಸೊ gametes are R, capital R and small r, here capital R and small r. So it forms R and R, capital RR dominant, right? Then with this capital R and small r, it forms capital R and small r. This is also considered as dominant. ಒಂದು capital letter ಇದ್ರೂನು ಅದು dominant ಅಂತಾನೆ ಹೇಳ್ತೇವೆ. But we have to differentiate, that is homozygous dominant and heterozygous dominant anta heltave. But still these two are the dominants. Then again this capital R is crossing with small r, it gives capital R and small r. Then again this small r and small r crossing gives two small r's. So this indicates the homozygous recessive. The small letter is always recessive, right? So ಇಲ್ಲಿ ಫಸ್ಟ್ ಏನ್ ಮಾಡಬೇಕು ಅಂದ್ರೆ ಫ್ಲವರಿಂಗ್ ಪ್ಲಾಂಟ್ ಅನ್ನ ಕೊಟ್ಟಿದ್ದಾರೆ, ದಟ್ ಈಸ್ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಆರ್ ರೆಡ್ ಫ್ಲವರ್, ಇಟ್ ಕ್ರಾಸ್ ವಿತ್ ಸ್ಮಾಲ್ ಆರ್ ಆರ್ ವೈಟ್ ಫ್ಲವರ್. ಸೊ ಇಟ್ ಗಿವ್ಸ್ ದ gametes capital R and small r. This is the F1 generation. ಇದು F1 generation. Next F2 generation ನಲ್ಲಿ capital R and small r is crossed with capital R and small r. So this is indicated by this column. ಇದು ಚೆಕ್ಕರ್ ಬೋರ್ಡ್ F2 generation. ಇದು ಕ್ವಶನ್ ಅಲ್ಲಿ F2 generation ಚೆಕ್ಕರ್ ಬೋರ್ಡ್ ಅನ್ನ ಕೇಳಿದ್ದಾರೆ. So now you have to write the phenotypic and genotypic ratios. How to write this phenotypic ratio? It contain how many dominant and how many recessive are comes. So ಇಲ್ಲಿ ಚೆಕ್ಕರ್ ಬೋರ್ಡ್ ಅನ್ನ ಫಾರ್ಮ್ ಮಾಡಿ ಆದ್ಮೇಲೆ ಇದರಲ್ಲಿ ಎಷ್ಟು ಡಾಮಿನೆಂಟ್ ಇದೆ ಎಷ್ಟು ರಿಸೆಸಿವ್ ಇದೆ ಅನ್ನೋದನ್ನ ಇಲ್ಲಿ ಏನ್ ಮಾಡಬೇಕು, ರೇಷಿಯೋ ಹಾಕಿ ಬರಿಬೇಕು. ಸೊ ಸಿ ಹಿಯರ್ 1, 2 and 3, all these three are the dominant, right? Because capital letter ಒಂದಿದ್ರೂ ಕೂಡ ಅದನ್ನ ಡಾಮಿನೆಂಟ್ ಅಂತ ಹೇಳ್ತೇವೆ. ಸೊ 3 dominants and only 1 recessive. So this is the phenotypic ratio. But right, for genotypic ratio ಅನ್ನ ಬರೆಯುವಾಗ ಇದನ್ನ ಸಪರೇಟ್ ಆಗಿ ಬರಿಬೇಕು. Here homozygous dominant, heterozygous dominant and homozygous recessive ಅನ್ನ ಡಿಫರೆಂಟ್ ಆಗಿ ಬರಿತೇವೆ ಇಲ್ಲಿ. So this capital R and capital R is 1. This is the homozygous dominant. Then capital R and small r, capital R and small r, 2 are present. So this is 2. This represents the dominant only, but heterozygous dominant. Then again only one homozygous recessive are present, that is small r and small r. So this is 1. So this is the genotypic ratio. This way we have to write the, or we have to form the checkerboard, and on the basis of that checkerboard you have to write the phenotypic and genotypic ratios. Thank you. Hello everyone. Let us see question number 36. Name the hormones that perform the following functions in humans. First one, controlling the sugar level in the blood. So ಶುಗರ್ ಲೆವೆಲ್ ಅನ್ನ ಬ್ಲಡ್ ಅಲ್ಲಿ ಶುಗರ್ ಲೆವೆಲ್ ಅನ್ನ ಕಂಟ್ರೋಲ್ ಮಾಡೋ ಹಾರ್ಮೋನ್ ಯಾವುದು ಅನ್ನೋದು ಕ್ವಶನ್. ನೇಮ್ ದಿ ಹಾರ್ಮೋನ್ ವಿಚ್ ಕಂಟ್ರೋಲ್ ದಿ ಶುಗರ್ ಲೆವೆಲ್ ಇನ್ ಬ್ಲಡ್. We know that our body contains so many glands, right? And each gland, it produces or it generates different hormones. So the sugar level, controlling sugar level in blood, this is the function of the hormone, that is insulin. Insulin anno hormone, it controls the sugar level in the blood. So this is generated or produced by the gland that is called pancreas, right? Pancreas ಇಂದ ಇನ್ಸುಲಿನ್ ಅನ್ನೋ ಹಾರ್ಮೋನು ಬಿಡುಗಡೆ ಆಗತ್ತಾ. ಈ ಇನ್ಸುಲಿನ್ ಅನ್ನೋ ಹಾರ್ಮೋನು it controls the sugar level in the blood. In the 36th question, in that second one, prepare the body to deal with emergency situations. Which hormone controls the body to deal with the emergency situations? Emergency situations ಅಲ್ಲಿ ಹೇಗೆ ನಾವು ರಿಯಾಕ್ಟ್ ಮಾಡಬೇಕು ಅನ್ನೋದನ್ನ ಯಾವ ಹಾರ್ಮೋನು ನಮ್ಮ ಬಾಡಿಯನ್ನ ಪ್ರಿಪೇರ್ ಮಾಡುತ್ತೆ ಅನ್ನೋದು ಕ್ವಶನ್. The answer is adrenaline hormone. Okay, this is generated by adrenaline gland. Adrenal ಅನ್ನೋ ಗ್ಲ್ಯಾಂಡ್ ಇಂದ adrenaline ಅನ್ನೋ ಹಾರ್ಮೋನು ಬಿಡುಗಡೆ ಆಗತ್ತ. This adrenaline hormone helps to prepare the body to deal with the emergency situation. See, this is the adrenaline gland, right? This is adrenal gland. It produces adrenaline hormone. Adrenaline ಅನ್ನೋ ಗ್ರಂಥಿಯಿಂದ adrenal ಅನ್ನೋ ಹಾರ್ಮೋನು ಪ್ರೊಡ್ಯೂಸ್ ಆಗತ್ತ. This hormone helps to prepare the body to deal with the emergency situations. And 36th, in that third question, controlling the fats, proteins and carbohydrates metabolism. Which hormone controls the fats, proteins and carbohydrates metabolism? That is thyroxine hormone. This is produced or generated by thyroid gland. ಥೈರಾಕ್ಸಿನ್ ಅನ್ನೋ ಹಾರ್ಮೋನ್ ಅನ್ನ ನಮ್ಮ ಥೈರಾಯ್ಡ್ ಗ್ಲ್ಯಾಂಡ್ ಉತ್ಪತ್ತಿ ಮಾಡತ್ತ. This hormone is the responsible for controlling fats, proteins and carbohydrates metabolism. Thank you. Hello everyone. Hello everyone. Let us see 15th main. Answer the following questions. In that question number 37, draw a diagram showing the schematic sexual sorry. I will repeat this question one more times. Hello everyone. Let us see 15th main. Answer the following questions. In that question number 37, draw a diagram showing the schematic sectional view of human heart. Label the following parts. First one, septum and right atrium. So this is the question asking for 4 marks. Here you have to draw the diagram of heart and labeling these two parts. First one is septum and second one is right atrium. Let us see how to draw this diagram. See, this is our beautiful human heart structure. Human heart. In question, they asked to label right atrium and septum. Okay, this is the right atrium part and this is the septum. So you have to label these two parts and draw this picture neatly. It contains 4 marks. Thank you. Hello everyone. Let us see 16th main. Answer the following questions. Question number 38 in that A. List the 3 methods of contraception that are usually followed by human beings. What are the methods of contraception that are followed by human beings? Here you have to write the 3 methods. In that first one, mechanical method. This is the first method of contraception, means using condom or using copper T. In second method, that is oral method, means consuming hormone imbalance pills. This hormone imbalance pills, it just imbalances the hormone in the male reproductive system and it stops the pregnancy. Then third method for contraception is surgical procedure. It is nothing but blocking vas deferens in male and blocking fallopian tube in female. This is also one of the method of contraception that are followed by human beings. So you have to write the 3 methods. So in that 38th B, what is the function of the vas deferens and prostate gland in the human male reproductive system? The question is, write the function of vas deferens and prostate gland. These two are the reproductive system in male. These two are male reproductive systems. You have to write the function of these two. So this is vas deferens and this is prostate gland. The function of this vas deferens is it transports sperms from tests. It transports sperms from tests. That is the function of vas deferens. So second one, what is the function of prostate gland, right? So this prostate gland is also the male reproductive system part. The main function of this is it provides the fluid medium and nutrients necessary for the transportation of sperm. So the main function of prostate gland, this gland provides fluid medium and nutrients for the transportation of sperm. This is the function. Then 38th C, how does the menstrual cycle occur? Explain. This 38th question having A, B, C sub questions. In that C, how does the menstrual cycle occur? Explain. See, this is uh, the female reproductive system. This contains ovaries, left and right ovaries. Due to this ovaries, every month one egg is released. If suppose it is get fertilized, then pregnancy forms. Otherwise, it is not fertilized, menstrual cycle occur. Now you have to explain it in words. If the egg is not fertilized, it lives for about 1 day. This ovaries which produces eggs in every month. If the egg is not fertilized, it lives for about only 1 day, that is 24 hours. Then thick and spongy lining of the uterus slowly breaks and comes out through the vaginal as blood as mucus. So, if suppose this egg is not fertilized in 24 hours, after that it converted as thick and spongy lining of the uterus slowly and breaks and comes out through the vagina as blood and mucus. This is called menstrual cycle. So this way the menstrual cycle occur. Thank you. Hello everyone. Let us see question number 38. This is OR question. In that A, again this 38 OR question contain sub questions A, B, C. In that A, write any two differences between self and cross pollination. You have to write the any two differences between the self and cross pollinations. This pollination is the process takes place in flowers, that is called asexual reproduction method in flowers, right? Let us see the difference one by one. First difference is in self pollination, it transposes the pollen from stamen to stigma of the same flower. But in cross pollination, it transfer of pollen from stamen to stigma of another flower. This is the first difference. See, in self pollination, this is just refer this diagram. This shows the self pollination. Here, the transfer of pollen from stamen. This is male reproduction part in flower. This transfer of pollen from stamen, right? This pollens from stamen part, it transferred to the stigma. This is the stigma part. This is called female part. Okay. It takes place in the same flower. ಒಂದೇ ಫ್ಲವರ್ ಅಲ್ಲಿ ಪಾಲನ್ ಸ್ಟೆಮನ್ ಇಂದ ಸ್ಟಿಗ್ಮಾಗೆ ಹೋಗಿ ಸೇರತ್ತೆ. ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್. ಬಟ್ ಇನ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್, ಟ್ರಾನ್ಸ್ಫರ್ ಆಫ್ ಪಾಲನ್ ಫ್ರಮ್ ಸ್ಟೆಮನ್ ಟು ಸ್ಟಿಗ್ಮಾ ಆಫ್ ದಿ ಅನದರ್ ಫ್ಲವರ್. ಇಲ್ಲಿ ಒಂದು ಫ್ಲವರ್ ಅಲ್ಲಿ ಇರುವ ಪಾಲನ್ ಗ್ರೇನ್ಸ್ ಇನ್ನೊಂದು ಫ್ಲವರಿನ ಸ್ಟಿಗ್ಮಾಗೆ ಹೋಗಿ ಸೇರಿ ರಿಪ್ರೊಡಕ್ಷನ್ ಆಗತ್ತೆ. ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್. ದಿಸ್ ಇಸ್ ದಿ ಫಸ್ಟ್ ಡಿಫರೆನ್ಸ್. ದೆನ್ ವಾಟ್ ಇಸ್ ದಿ ಸೆಕೆಂಡ್ ಡಿಫರೆನ್ಸ್? In this self pollination, media is not necessary, but cross pollination, it requires some media like air, water, bird, insects is the necessary media for cross pollinations. So in self pollinations, media is not necessary. Means self pollination ಒಂದೇ ಫ್ಲವರ್ ಅಲ್ಲಿ ಆಗೋದ್ರಿಂದ ಅದೇ ಫ್ಲವರ್ ಅಲ್ಲಿ ಇರುವ ಸ್ಟೆಮನ್ನು ಅದೇ ಫ್ಲವರ್ ಅಲ್ಲಿ ಇರುವ ಸ್ಟಿಗ್ಮಾದ ಜೊತೆ ಸೇರತ್ತೆ. ಸೊ ಅದಕ್ಕೆ ಬೇರೆ ಮೀಡಿಯಾದ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಬಟ್ ಇನ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್, ಇಲ್ಲಿ ಒಂದು ಫ್ಲವರ್ ಇಂದ ಇನ್ನೊಂದು ಫ್ಲವರಿಗೆ ಹೋಗಿ ಸೇರ್ಬೇಕು. ಸೊ ಒಂದು ಫ್ಲವರಿನ ಸ್ಟೆಮನ್ ನಲ್ಲಿರೋ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಇನ್ನೊಂದು ಫ್ಲವರಿನ ಸ್ಟಿಗ್ಮಾಗೆ ಹೋಗಿ ಸೇರೋವಾಗ ಇಲ್ಲಿ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡೋದಿಕ್ಕೆ ಮೀಡಿಯಂ ಬೇಕಾಗತ್ತೆ. ಸೊ ಮೀಡಿಯಂ ಈಸ್ ನೆಸೆಸರಿ ಫಾರ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್. These two are the differences we can write for self and cross pollinations. Then 38th B, what are the changes that occur in flower after fertilization? So after fertilization, what are the changes we can observe in flower? That is the question. Once the fertilization takes place, there is so many changes takes place in flowers. Let us see it step by step. Once the fertilization occur, after this fertilization, the zygote divides several times inside the egg and producing an embryo. See, once the fertilization occur in flower, the zygote, it divides several times. cell division takes place here inside the egg and it producing an embryo. So once the embryo is formed, the petals, sepals, stamens, pistils and stigmas fall off easily. So these all are the remaining part of the flower. Petals, sepals, stamens, pistils and stigmas all are fallen down like this. Then the egg develops a thick layer that slowly changes into a seed. First of all, after fertilization, the zygote divides several times inside the egg and it produces an embryo. After this, the petals, sepals and remaining part of the flowers slowly fall off easily. Then the egg develops a thick layer that slowly changes into a seed. The ovary grows rapidly, matures and repines. So this is the process takes place after the fertilization in flower. These all are the steps that includes the changes takes place in flower after the fertilization. Let us see 38th question in that C. Mention any 4 advantages of vegetative propagation. What are the advantages we can see in vegetative propagation method? You have to write at least 4 advantages here. So first advantage is they produce flower and fruit quickly. As compared to other method, this vegetative propagation gives the product so fast. It produces flower and fruit quickly as compared to other method. So this is the first advantage. And second advantage is this method is useful in plants that do not produce seeds. So in some plants they do not produce seeds. So that plants uses this vegetative propagation is very helpful because this is helpful for the plants that do not produce the seeds. ಯಾವ ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಸೀಡ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗೋದಿಲ್ವೋ ಆ ತರಹ ಪ್ಲಾಂಟ್ಸ್ ಗೆ ವೆಜಿಟೇಟಿವ್ ಪ್ರೊಪಗೇಷನ್ ಮೆಥಡ್ ಅನ್ನೋದು ತುಂಬಾ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ವಿತೌಟ್ ಸೀಡ್ಸ್ ಇಲ್ಲಿ ನಾವು ಏನ್ ಮಾಡಬಹುದು, ವಿ ಗೆಟ್ ಫ್ಲವರ್ಸ್ ಅಂಡ್ ಫ್ರೂಟ್ಸ್ ಅಂಡ್ ಆಲ್. Then the one more advantage is they produce high yielding disease free plants. ಇದು ಏನ್ ಮಾಡತ್ತೆ ಅಂದ್ರೆ ಬಹಳ ಬೇಗ ಬೆಳೆಯನ್ನ ಕೊಡೋದ್ರ ಜೊತೆಗೆ ಡಿಸೀಸ್ ಫ್ರೀ ಇರೋ ಪ್ಲಾಂಟ್ಸ್ ಅನ್ನ ಕೊಡುತ್ತೆ. This is also one of the advantage of vegetative propagation. And fourth advantage is a large number of plants can be grown in less time, less space and at lower cost. This is also one of the major advantage. Here large number of plants can be grow in a less time. ತುಂಬಾ ಕಡಿಮೆ ಟೈಮ್ ಅಲ್ಲಿ ಅತಿ ಹೆಚ್ಚಿನ ಬೆಳೆಯನ್ನ ಬೆಳಿಬಹುದು. ಹಾಗೆ ಇದಕ್ಕೆ ಸ್ಪೇಸ್ ಕೂಡ ಕಡಿಮೆ ಸಾಕು. ಅದೇ ರೀತಿ ದಿಸ್ ಇಸ್ ಬಜೆಟ್ ಫ್ರೆಂಡ್ಲಿ, ಅಂದ್ರೆ ಲೋ ಕಾಸ್ಟ್ ಅಲ್ಲಿ ಕೂಡ ವೆಜಿಟೇಟಿವ್ ಪ್ರೊಪಗೇಷನ್ ಅನ್ನ ಸುಲಭವಾಗಿ ಮಾಡಬಹುದು. ದೀಸ್ ಆಲ್ ಆರ್ ದ ಅಡ್ವಾಂಟೇಜಸ್ ಆಫ್ ವೆಜಿಟೇಟಿವ್ ಪ್ರೊಪಗೇಷನ್. ಅಟ್ ಲೀಸ್ಟ್ 4 advantages you have to write. Okay, thank you.